All right. Hi, everyone. Sorry, I am a little late getting this up, but I'm going to try to actually get as much of the chapter covered in this recording so that way we don't have to cover as much and we can maybe host a little bit of a review session on Monday. So that way you're not covering new content, hopefully Monday, and also then you can use Monday to rewatch this lecture or whatever. So again, a couple of little shop things. We're going to be covering chapter three. I'm going to try to get through all of chapter three before our exam on Wednesday. Keep in mind when it's time for the exam, it is multiple choice and short answer. You do need to bring a Scantron. It's going to be in person on campus in our class time, so make sure that you're on time. I'm trying to think if there's anything else you need to know for our lecture, for our test. Um, I'm sure I'll think of it, but I'll do more announcements and things as we get a little bit closer. Um, Keep in mind, the sooner you turn in your study guide, if I have time this weekend, I'm trying to review them and give you guys feedback on stuff that maybe you might have wrong or that you should go a little bit deeper. If I suddenly get an influx of everyone's study guides all at once, then I won't be able to do as much feedback. So even if it's only partially done or you have some questions, I've had some students already email me and I'm trying to get back to everyone over the course of the weekend. So don't hesitate to ask me questions. Bring your study guide with you Monday if you want to ask new questions. Like I said, I want to host some time to get you feeling comfortable and ready for the test. I am going to do a little mini pop quiz, our first pop quiz on Monday, and I'm letting you know today so that way those of you who listen to this recording, you will be prepared, and then we'll kind of know those that don't listen to the recording as well. Um, so you're going to have two questions. This is not going to be open book, open note. I want you to do the best that you can. Again, anyone who writes anything down for this is going to get three-fourths credit because this is a quiz for attendance, but you might lose a point or two just out of the 10. I think they're worth 10 points each, if I'm not mistaken. You might lose a point or two if you get the questions completely wrong. So your questions on Monday, so that way I hope people actually come and listen to this recording. Your questions on Monday are going to be define or describe homeostasis. And describe, I want you to describe as many of the properties of water as you can. So in our exam, I may not have you do all 5 or 6 properties of water, but for our pop quiz on Monday, I really want you to know as many of the unique, important properties of water that you can remember. So rewatch that crash course video. I think that the crash course video I gave you is the best kind of layout of those properties of water. And then review the book and the sections in the book. There's either 5 or 6. I'd have to go back and look, but either way, just try to remember as many as you can. Oops, I'm getting a bunch of little pop-up things. Okay, so you have a pop quiz Monday, define homeostasis, and describe as many of the properties, unique properties of water as you can. Those are going to be your two questions. It will not be open book, open note. I'll give 8 or 9 minutes at the beginning of class. I'm going to try to get there early so I can have it up on the board and you can start it as soon as you arrive. But either way, you'll have 8 or 9 minutes to get that done. All right, so let me share my screen and let's get going on chapter three. This is not going to be super interesting. I don't have a lot of extra materials because I kind of want to knock this out. And then we'll kind of reset after the exam next week. So let me share my screen. Let's go over there. All right. Oops, that's the wrong one. Well, you're seeing some questions. Stop sharing. I thought that was my PowerPoint. All right. Let's share my screen. Let's find the actual right one. All right, there's chapter three. So this is taking our chemistry information that we started with and expanding on that and making it, actually, we're narrowing it down and making it more relate to what we're going to be talking about in life sciences and biology. Let me go into that mode. So we're discussing now the molecules of life. We've talked a little bit about atoms. We've talked about the different components of atoms. And now we're going to talk about how this chemistry relates to biology and life sciences. So in this case, we're going to talk a bit about carbon. And carbon is kind of our life giver, right? So the chemistry of biology, we're discussing carbon. Carbon is uh, it's an atom. It's on our periodic table of elements. It's an element. It's our periodic table of elements. It's represented by a C. And they are molecules that have primarily hydrogen. Oh, stuff of life specifically is molecules that have hydrogen and carbon, but carbon is the one that we kind of hear about all the time when we're relating to the term organic. Now, we're not necessarily talking about organic like we do with our foods in the store. We're talking specifically about the term that typically relates to the presence of carbon in a molecule. Carbon is incredibly important because it's incredibly versatile. It has four vacancies or spaces where those electrons can be shared. So it has room to share and bring in electrons to make those ionic and covalent bonds. Um, and so most of our organic molecules are going to have something called a backbone, a carbon backbone, and it's literally shaped like a little backbone or a chain of these rings of carbon. This is what we're talking about when I say a ring of carbon. Now, this is a true ring, and each C that we see represented here is an actual carbon atom, and then they're linked together and they're sharing those electrons to make this ring shape. So again, it gives rise to life in so many ways, and it supports life in so many ways because it assembles, it's so versatile, it assembles into lots of compounds or different molecules, right? There's something called a hydrocarbon, which is going to be just carbon and hydrogen. It also forms into something called a functional group. This will come up a bit more later, but a functional group is an atom other than hydrogen bonded to a carbon. So it's going to have, it bonds to these other groups. It allows for these little links to be made to other groups that start to give us new um, molecules. And again, we'll get into those specifics here in a little bit. So biological molecules are often modeled to reduce some complexity. So we're just, everything is about models. We're always trying to find ways because this is obviously not visible to the naked eye, so we try to find ways to represent that, like in this case. So we're talking about a functional group. You should not need to memorize these, but I do want you, if I'm going to go back through your test, you shouldn't, I wouldn't expect you to memorize all of these. I might want you to know what a functional group is. So back here, it's an atom other than hydrogen that's bonded to a carbon. And they all have these terms, amine, you'll see here in the middle, or methyl, acetyl. These are all ketones. These are all things that maybe you've heard if you're in the nutrition space at all. Maybe you've heard of acetyl or aldehydes, things that have to do with alcohols or sugars or ketones. All of these things are terms that you've heard, and it has to do with their bond to these carbon groups. So you'll see here a methyl group at this very first one. There might be a carbon ring, right? We saw a couple slides ahead, or a carbon chain, a carbon backbone. And then we have this little group coming off, and so it's a carbon attaching to a carbon with those three hydrogens around it. And it's literally represented as a formula for those of you who aren't as familiar with chemistry. This is a chemical formula for a methyl group. So if you look, there's a single C and three H's. You see that in the structure, and you also see that here in the formula. So look under the word structure, you'll see C with three H's, and then in the formula, it's literally listed as CH3. So they try to keep it as simple as possible. And when you start getting into this organic chemistry, which we're not doing very deep, but again, that is life chemistry, right? You'll see here this hydroxyl group, OH group. We talked a little bit about OH groups, or hydrogen, when it came to our pH. And when we talk about hydroxyl groups, this has stuff to do with that if there's more OH or H plus. But I won't expect you to memorize that. So this is just some ways that they're recommended, and those functional groups can be found in these different compounds. So like the methyl group is found in fatty acids, hydroxyl groups are found in alcohols or sugars, carboxyl groups are found in fatty acids and amino acids, and so on. And then here's those models that we mentioned. These are all different representations. If you go as far as organic chem, which I don't think most of you will need to, but if you did, you'd do a lot with this letter A here, where you're drawing all the little links with the C's and the H's around them. But just know these are different models or representations of different organic molecules. So why do we care? Why do we care about carbon? We know that it's versatile. We know that we use it a lot. Obviously, it's used in simple sugars, proteins, nucleotides, DNA, RNA. Obviously, there's a use for it. But something that we talk about a lot is metabolism or metabolic reactions. So this is the part where I think we started to get into in your study guide a little bit that we hadn't quite covered yet. But here we're going to talk about these metabolic reactions. So all biological systems are based in the same organic molecules. So that's going to be the carbon, hydrogen, oxygen, and nitrogen. There's going to be different amounts, and how they work and what they do will be different depending on the organism that we're talking about. But all of them are going to have this same organic molecules. So there's the terms that were listed in your study guide. We have monomers, which are subunits of larger molecules. So a monomer is a generic term, and we're going to get into the specific terms, but a monomer is just a subunit. We could be talking about sugars, a single sugar that links into other sugars. A really common one that we talk about is like cholesterol. It starts as a monomer and then builds into polymers. So get used to those prefixes: mono, one; poly, more than one. And all of these things build, right? We have simple sugars and we have complex carbohydrates, right? We have fatty acids, and then they build into other substances. So we'll go through here and we'll get into those specifics as we go. So cells build polymers from monomers, and they break down polymers. If we take in a polymer, then we can break it down to release monomers. These are all called chemical reactions, and this is the essence of metabolism. So metabolism is are chemical reactions, lots of chemical reactions where cells acquire and use energy. So obviously we think about taking in a fuel source. There's a picture of a donut at the beginning. We take in that donut. That's now a fuel source. It's made of sugars and fats and all kinds of things, but they're built into this whole donut. So we want to access the fuel in them. And so we'll break down the different parts of the donut. So we're breaking down these polymers into monomers to use the energy, to acquire and use that energy. And then there's a term called an enzyme, and that's going to be an organic molecule that just speeds up reactions. And this goes deeper into chemistry, but just know what enzymes are. They are super helpful with making sure that we don't fatigue our bodies. We have enzymes all throughout our metabolism and other systems. Some other terms to know: hydrolysis is a chemical reaction where an enzyme uses water to break a molecule into smaller units. So hydrolysis is adding water to break bonds. Okay, so it's a hydrolysis, hydro, water, lysis, breaking or killing, breaking. And so water breaking, we're sending water in to break bonds, to break up a polymer into something more simple. There's also condensation. That's a chemical reaction in which an enzyme builds a large molecule from smaller subunits. Let me see real quick. I want to make sure that I'm even making any noise. Um, okay. Hold on. I'm just making sure that my audio is working. I got paranoid real quick. All right, back to where we were. Sorry for that interruption. Um, Let me reshare. Okay. Sorry about that. I panicked for a moment that I was recording for the last few minutes and then that it wasn't actually recording. So we'll continue. So condensation, we said hydrolysis. Condensation is a chemical reaction in which an enzyme builds a large molecule from smaller subunits. So water is formed during condensation. That's going to be our main topic of condensation. But this is it can be used in other molecules. So enzyme builds larger molecules from smaller ones. Hydrolysis breaks complex ones into simple ones. Condensation takes simple ones and adds to them. Here are some metabolic reactions. So carbohydrates. Carbohydrates, now this is a part that gets where we are now taking our term monomer and polymer, and we're gonna address the different larger groups that we're gonna start discussing. Carbohydrates being the first. They have their own terminology associated with their singular and multiple parts. So a carbohydrate is an organic compound that consists of carbon and hydrogen. So now we're talking about an actual usable compound, something that we're going to take in and consume and potentially use fuel from. So carbon, hydrogen, and not oxygen in a 1 to 2 to 1 ratio. So one carbon, two hydrogen, one oxygen is the ratio. There are three types of carbohydrates in living systems. So this is going to start looking familiar but new. So we have monosaccharides, oligosaccharides, and polysaccharides. We primarily talk about mono and polysaccharides, but just go with those prefixes. But now we're seeing saccharide, and that secondary part of that word is relating to sugar. So try to get used to saccharin, saccharide, these terms that start to have meaning like saccharide relating to sugar. Monosaccharides are a single sugar and simplest type of carb. They will have a carbon that has, you saw those carbon rings. So it'll either have 5 or 6 carbons in that ring. So when we're talking about glucose, so right, we're talking about sugar, that one of the sugars we consume regularly. It has 6 carbons. 5 carbon monosaccharides are going to be part of DNA and RNA, and those we'll get into at a later lecture as well. So these are what cells, monosaccharides are used for fuel. Breaking the bonds of sugars releases energy. So again, we have all this, those are nicely shared, right? We said they have 4 places and they have 4 vacancies to share those electrons. Then we can break those bonds, and when we break them, there's a release of energy. And so then the cells can undergo metabolism. They can by breaking these bonds and utilizing that energy. Monosaccharides can also be used as precursors for other molecules and as structural materials. So here we can see this is a 5 carbon sugar called ribose. And at the very top here, I know it's hard, I'm not able to point, but at the very top where that O is kind of embedded in that carbon, just know there's a carbon there right at the point. Each point represents a carbon molecule, and then it's also attached to an oxygen molecule. Then you can see that there are functional OH functional groups that are coming off from each additional carbon. Remember that the 5-point carbon is going to be what goes into our DNA and RNA. Then we have short chain carbohydrates. So these are going to be disaccharides, two sugars, which is two monosaccharides bonded together. That's going to make something called lactose and sucrose. That's going to be made of, so we have our glucose sugar plus a galactose sugar or a fructose sugar. You don't have to memorize all that. Just know that these are, just understand. You don't even have to know necessarily. But just understand that disaccharides are also usable. We hear the word lactose all the time. Probably half or more in class are lactose intolerant. So those are terms hopefully that you're used to. And now you know that those are two sugar um, short chain carbohydrates. And this is kind of what they would look like. And they're bonded together. Then we have polysaccharides. Polysaccharides, again, several or many sugars, chains of hundreds or thousands of monosaccharides all together. The most common ones now, these are going to be stronger, tougher. These carbohydrates might serve different purposes, but they are still polysaccharide sugars. There's a sugar or carbon component, and that's going to make cellulose, starch, or glycogen. Cellulose, and I do want you to kind of know what some of these are. Cellulose is the main structural component of plants. It is insoluble, meaning that we're not going to be breaking too many bonds from that and harnessing the energy. It's going to be composed of glucose monomers stretched side by side and hydrogen bonded. So it's going to be tough and insoluble. It's the bones of plants. It's what keeps them upright, what helps them keep their form. Then there's starch. This is going to be the energy reserve in plants. So you think of a potato, it's all full of starch. That's lots of energy, but it's not active energy, right? It's being stored in that potato just as a nice neutral, nothing's happening. It's just there in case the plant would need it or then for others to consume and utilize those starches. So it's the main energy reserve in plants. It's stored in roots, stems, leaves, seeds, and fruits. And it's got a bunch of glucose monomers that form a chain that coils. You don't have to know all of those, but I would like you to be aware that cellulose is the structural part of plants, starch is the energy reserve in plants, and glycogen is the energy reserve in animals. This might be one that you're less familiar with, unless maybe you have a family member that talks a bit about their diabetes, or you've taken kind of a health science class before. This is one of those big terms that comes up. So that's a main energy reserve in animals that includes us. It's very abundant in muscle and liver cells, and it's lots of glucose monomers with lots of chains. So here's our three types. You can see in the top left here, A, that's going to be that cellulose. B is going to be the starch. And C is going to be the glycogen. Our next kind of group of molecules is going to be lipids. This also has their own terms, their own monomers and polymers that have their own terms. Lipids are fats. Try to get used to thinking that, tell yourself that, flashcard that, whatever else. Lipids are fats. They can be oily, they could be waxy, but they will be some kind of fat. Many lipids incorporate fatty acids, which is going to have something called a hydrocarbon, right? Water, carbon, or hydrogen carbon tail with something called a carboxyl functional group head. Get used to those terms, hydrophobic and hydrophilic. This is going to come into play when we talk about cells. So these lipids are what form the outer barrier to all of our cells. And we have to get used to remember that the head is hydrophilic. It loves water. The heads of these hydrocarbons love water. The tails of these hydrocarbons are afraid of water. And that's what starts building that outer boundary of our cells. Um, when it comes to kind of consumption, we can talk a little bit about lipids in living systems. So, saturated fatty acids have single bonds linking the carbons in their tails. Here we can see the one with the actual like brackets is a saturated fat. And then unsaturated fatty acids have double bonds linking them. So if you look at the difference here, because I can describe that to you, but it's much easier to see. This one on the left-hand side, the stearic acid, letter A, that's considered saturated. It's very straight. Every single carbon has two related hydrogens, and then whatever's on the end has some extras. But every single carbon is identical. They create this, they're fully saturated with hydrogen, and that's what that means. Whereas these others, B and C, our linoleic acid, are unsaturated, and you see that some of our carbons are double bonded, and therefore they're not saturated with hydrogen. And that's the difference between what we're talking about when we're saying saturated fats and unsaturated fats. Fats specifically are lipids that consist of something called a glycerol molecule. Again, they take different forms, but fats contain glycerol. So then we talk about, if you've ever had blood work done, this concept of triglycerides. It's a fat with three tails, three fatty acid tails. So saturated fats are triglycerides with those with saturated fatty acid tails, and they're going to be solid at room temperature. Unsaturated fats are liquid at room temperature, and they're going to have unsaturated tails. So here is a triglyceride, and we said saturated. I don't like that picture for you guys, but we have our triglyceride, three fatty acid tails, and that's just these little zigzags. Again, I haven't gone into all of the organic chemistry, but at each point of the zigzag is another carbon molecule. So just know when you're drawing it in a ring or a zigzag, carbon is what's being represented at those points. And it's just a space saver to not like write a C at every single point. So this is where we get into, again, life, cells. How do we have cells to begin with? We know that oils, fats, and waters don't mix. And so having access to these lipids, these what are now we're going to use the term phospholipid, and you're going to hear that a lot throughout the rest of class. So get used to the term phospholipid. It's going to be the main component of a cell membrane. So because fats and waters don't mix, then they're able to separate and they're able to keep this boundary now. So we're going to talk about these phospholipids. That's what makes a cell membrane, or the outermost boundary of a cell, and it contains a phosphate group in hydrophilic head, right? So again, the water-loving head has a phosphate group, and then those fatty acid tails, kind of like what we saw above, those fatty acid tails are going to be hydrophobic. And this is very important for our future lectures. So that's why I want to really take this concept of phospholipids home. I would say expect a little pop quiz on this topic, even if it's after the exam. I may cover this again because I want you to understand phospholipids. I don't think I have too many questions on the exam related to this. If I didn't put it in the study guide, it's not there. But phospholipids come up a lot later. So phospholipids make cell membranes. They are arranged in a way that makes a ring, and it's going to have them facing each other, facing away from each other. And so they're going to be a phospholipid head at the top and a phospholipid head at the bottom, and they come in pairs. So they make this kind of paired circle. I want to actually they didn't give us a good photo. If you guys remind me in class, I'll show you a much better video so you can see what I'm talking about. But just know we're not really going to see that many phospholipids loose and by themselves. They're going to hang out with a buddy, okay? So there's one layer of the hydrophilic heads are going to be on the interior, and then on the exterior, they're going to have another set of heads. I hate the way they've described this. So if you look at this photo here on the right, so this is a good-ish picture. Um, Here you see a phospholipid. I just want you to look at letter B. There's a phospholipid bilayer. Those tails are pointing towards each other. Hydrophobic tails are pointing towards each other, and hydrophilic heads are on opposite sides. One is going to be inside the cell, and one is going to be outside the cell. So one is inside and it's touching all the water and liquid on the inside of the cell, and one is on the outside, and that's going to be in the outer extracellular space. Um, so I do want you to know the term hydrophobic and hydrophilic. I do want you to understand that they come in bilayers and that they are the essence of cells. They're what make cells, right? The component of cell membranes. Moving on to waxes. Wax is going to be now the kind of more complex structure, kind of like the cellulose in starches in carbohydrates. Wax is going to be complex, varying mixtures of lipids with long tails bonded to alcohols or carbons, and there's different kinds of waxes. The molecules will pack tightly, so waxes are actually water repellent. That's why if you think about even just candle wax or beeswax, the water beads up and just runs right off because they're firm. They're not going to allow water to escape through very easily. Plants will secrete waxes to restrict water loss. It's also a protective coating. Other types of waxes protect, lubricate, and soften skin and hair. I think I told you guys a little bit on the first day that like my study for grad school was ticks and then this kind of wax producing organ. So that's used to protect eggs. Waxes can be used in lots of different ways, often for protection or separation from water or something like that. Then we have steroids. These are going to be lipids with no tails. They will have a rigid backbone. The functional groups will define the type of steroid. The reason that we talk about them and the reason that it's important to get fat in your diet is because some examples of steroids are estrogen and testosterone. Obviously, we all have varying amounts of that in our bodies. And so we need fats to help have the monomers to build these polymers, right, of these steroids. Our next molecule is going to be protein. I feel like all I hear about, especially if you're a fitness person at all, we all are trying to get in as much protein as possible. So it's going to be proteins are made of amino acids. Their subunit, instead of like monosaccharides or whatever, these are going to be now amino acids. That's what makes proteins. Proteins also, especially if we get time to discuss that later in the semester, are the essence of who we are. Proteins are what make us us. They're what give us our hair color, our hair texture, our just even I'm just trying anything that makes us us. It's what makes our lungs be lung tissue. It's what may or may not give us freckles, or it's all of the things. So proteins are incredibly vital for differentiation amongst organisms. So they are made of amino acids. The cells can make thousands of different proteins from only 20 monomers called amino acids. An amino acid contains something called an amine functional group, a carboxyl functional group, and something called an R group. The R group is kind of also what determines what that protein will be. So you see we have our carbon here in the middle, hydrogen on top, a functional group on each side. So when we're building proteins, there's going to be covalent bonds that link those individual amino acids into a protein, they have their own bond. So now the terminology can get a little complicated. Um, the amino acids are linked via peptide bonds. A short chain of amino acids, so just a few of them, is called a peptide. As it gets longer, it's now a polypeptide. So that's going to be our terminology related to individual groupings of proteins. Proteins consist of polypeptides that can be hundreds or thousands of amino acids long. Um, Luckily for this class, you don't have to know all of those functional groups again. Uh, I'm surprised I didn't put ones that are more familiar. These are used. So we have something here called methionine or valine. These are the R groups. Again, we have the amine group, the carboxyl group, and then an R group. And that R group is what we're talking about when we're talking about these amino acids. Um, There's a whole list of them. We don't have to get into that. I'm not expecting you to remember the name methionine or anything like that. Just that amino acids use now some of these structures to create proteins. So something that is said a lot in biology is structure determines function. So how something is made is incredibly important and will determine its later function. Um, and sometimes one tiny little change in the structure of a protein could give it a completely different function. So a simple route is, let's say, it's a way too simplistic, but like hair color, uh, a slight change in that protein now means that we can have wildly different hair colors, right? From the blackest of black hair, blue black hair, all the way to maybe someone who has no pigmentation. So somebody that maybe their protein changed too much and now they have albinism and they have no color proteins in their hair or they're non-functional. So that little change in structure can have big changes in function later on. So proteins function in movement, defense, cellular communication, everything. A protein's biological activity arises from and depends on its structure. So structure determines function. The primary structure, so these are terms that come up, especially if you're in science, especially if you're in nursing, this will come up later too. But there's the primary structure. I hate when they don't just show us. But primary structure is linear. So it's like a straight line. It's just listing what the amino acids are. It'll tell us what kind of protein it is. Secondary structure. So this is when proteins are not just little lines of amino acids. They fold and touch and they start to interact and shape. And I have some videos from that too that I think will be helpful for you to see. But they'll change their shape. Um, so that way they interact, and certain parts are able for metabolism and cell structures and different things to read and express what we want to see, whether that's brown hair, blonde hair, freckles, no freckles, deeper voice, any of those things, right? So the folding is the secondary structure. We have a line for primary, folding and twisting for secondary. Tertiary is now these two proteins that are, or two edges of proteins that are starting to interact with each other. And quaternary structure is two or more polypeptide chains, and we almost never hear about quaternary structures. Don't worry about this definition. The main ones you need to know are primary. That's the name of it. It's nice and linear. We can see that here. This is primary, where it says methionine, valine, histidine, leucine. That's just primary. Then we see the number 3, and it's starting to fold into something called a helix, kind of like our DNA structure is. Here again is what our secondary structures would look like. Here on the left, the helix. Um, the other type of folding is this thing on the right called a beta pleated sheet. We don't have to know all those terms. A helix you should know because we're going to talk about DNA later. But this is a secondary structure, so still looks complicated. That's just how we represent secondary structures. Now we're talking about tertiary and quaternary. Let me see if I go back to the term domain. I know I saw it here. Tertiary structure. Yeah, so in tertiary structure, non-adjacent regions of protein join to create compact domains. So now it's not just that they're twisted, it's that they're actually folding in to interact with self and allow for other molecules to potentially open up space for them to interact with that protein. And then we have this quaternary structure. And that quaternary structure is two or more polypeptide chains closely associated or bonded. So now you're seeing we have what looks like about four proteins there, and they're all starting to interact. Enzymes often attach sugars or lipids to proteins, so they help speed up the process of attaching these different chains together. So as I said, and I'm going to stress to you guys over and over again, structure is function, the importance of protein structure, um, and how sensitive it can be to change, and how that can result in major problems in function. So some things that can affect protein structure. So you see how complicated these little spinning helixes are, right? And if one small change can cause drastic outcomes, we need to know what can cause those changes, especially if it's unwanted. So heat, salt, changes in pH. So now we're bringing back homeostasis into this discussion. Detergents, meaning soaps, can do something called denature or break down, unravel, change that structure by breaking hydrogen bonds. So they'll interfere with the hydrogen bonds on those carbons and other things. They'll break those hydrogen bonds and cause those proteins to unravel. And now they're not exposed and shaped in that perfect way so they can cause the correct function. Denaturation, and I want you to know that term, denaturation causes a protein to lose its function. Um, so they really threw this in here to scare everybody. So prions or prions are misfoldings of the glycoprotein PRPC, and this is now called an infectious protein. So we've heard a bit, I'm sure, obviously, we've all been through COVID, so you've heard a bit about viruses. Everybody should be familiar with the term viruses. So we know that bacteria are living organisms. Viruses are non-living, um, semi-non-living, so they require access to our cell metabolism in order to reproduce. And prions are neither of those things. So there's no actual living component, and so they're just infectious proteins. Um, there's probably the most famous one. They talked about it a lot in the 90s too. I feel like I always saw some scary late night Channel 5 special or 60-minute special on mad cow disease. Make sure you cook your meat, especially your wild game. Um, Make sure you cook it all the way through because if you cook it, it denatures those proteins and then they're no longer infectious. I actually do know recently it was someone who knew someone, they weren't close to me in any way, but someone my husband worked with, he actually passed away from getting mad cow disease. So it is still a thing that exists. And once you have it, we don't have any treatment for it. Um, it's not like a virus where we can at least try to use these kind of toxic antivirals. Well, not antivirals are toxic, but where we use, for bacteria, we can start using some, if it's a really resistant one, we still at least have really strong antibiotics to work against bacteria, and we have antivirals to help reduce viral loads, but we don't have anything to treat these prions. So they are pretty scary. And then if you're on TikTok at all, I feel like maybe it's just me because I'm on sciencey TikToks, but I hear about them way too much. So they are scary. I don't recommend looking too much into it. Just cook your meat well. We won't get into Creutzfeldt-Jakob disease. It's one that is can it's targets a specific community, specifically those who are of Jewish ethnicity. But we won't go too far into that. Our next group, let me see where we're at. Oh, we might actually just finish today. Let me see. Nucleic acids. I'm gonna finish this section on nucleic acids. Nope, that's gonna be it. We're done after that. So let's finish up here with nucleic acids. That's gonna be our last compound that we're gonna discuss. So we talked about our amino acids and proteins, and we've talked about our sugars, and we've talked about our fatty acids. So nucleic acids are made up of nucleotides. They consist of sugar with a 5-carbon ring. This is important for later when we get into DNA. So nucleotides have a 5-carbon sugar, right? Remember we said that's related to DNA and RNA. And it's bonded to nitrogen-containing base and 1, 2, or 3 phosphate functional groups. I want you to look at this term ATP, adenosine triphosphate. That's going to be an energy carrier. ATP is something you're going to see a lot throughout our cell discussions and then in any of your anatomy courses. Nucleic acids are chains of nucleotides in which sugar of one of the nucleotides is bonded to the phosphate group of the next. So nucleic acids are the multiple chains of are is a chain of nucleotides, right? So let me see, I fumbled over that. Chains of nucleotides in which the sugar of one nucleotide is bonded to the phosphate of the next. We'll see that here in an image, I'm sure. Yes. So nucleic acids are RNA, ribonucleic acid, or DNA, deoxyribonucleic acid. Both of those are going to be part of what tells our proteins what to do, right? DNA is our blueprint. It's our code. It's our code that makes us who we are. And RNA is one of the kind of interim, at least in humans and in mammals and stuff, RNA is um, one of the like messengers, templates. We'll get into all that. But RNA is single-stranded chain of nucleotides. DNA is a double-stranded nucleotide, chain of nucleotides. So we're looking here at nucleic acid structures. If you look at that orange ribose in the middle, it's got what, like we said, a phosphate group. That's going to be these 3 yellow P's on the left, and then a base, right? So this one has adenine. We will get deeper into that later. I don't think we're doing it in this one. No. So just know there's the phosphate group, the sugar backbone, and then the base on the blue side, the right side, which you can see also has its own set of carbons and hydrogens and all that. So A is going to be that RNA. It's a single strand. And then B is the double-stranded DNA. And if you think about our structures that we talked about earlier, these are representing that secondary structure, right? It's folded into that helix shape. So it's no longer a straight line. We now are seeing some folding and some interacting of those different groups. And just keep in mind, here it says the sugar of one nucleotide. So here we have a nucleotide. The sugar of one nucleotide is bonded to the phosphate of the next. We'll get into that as we get into more DNA info, but just look at the phosphate on the left is going to then connect to another sugar, which is going to connect to another phosphate, and that's how we build these structures. Here we have a little bit on, this is all just the extra stuff from let me see, yeah, from the lecture. So I'm going to go ahead and stop sharing. Hopefully you guys got through this. I am open for questions. We will probably review a chunk of this on Monday, but come with your questions. Remember, we're going to probably have a little pop quiz, so make sure that you're prepared for that. Again, I said it was going to be describe homeostasis and the properties of water. All right. So you guys are all set, and I will see you Monday. Take care. Oh, let me stop recording.